1984. Earl Washington Jr. is convicted for the rape and murder of Rebecca Lynn Williams and receives the death penalty. He goes on to serve 17 years in prison, but just a week before his execution, he is given a stay. 7 years later, it is shown through advanced DNA testing that he is not guilty, and he is finally pardoned in 2000. This story was just one of the many miracles of biotechnology. In fact, around the same time, we used it to sequence the entire human genome. And what I love about biology is that it all started from a simple red seaweed plant. Hi, today we're going to talk about gel electrophoresis, the topic which links all of these things together and is necessary for DNA profiling, DNA sequencing, and all sorts of modern DNA analysis. Gel electrophoresis is defined as the process which separates DNA fragments or even proteins, we'll come to that, but for now we're focusing on separating DNA fragments by their charge and their size. We'll come to why that's important, but first, how do we do it? The entire concept of gel electrophoresis is based on the simple fact that DNA has an overall negative charge. If you see on the left here, we have one half of a DNA molecule, that is to say one strand. We'll have another complementary strand attached to each base. But if you focus on the sugar phosphate backbone of the DNA, you'll see that each phosphate group has this one oxygen with a -1 charge. And this is because it has an extra electron. So when you add this all up, you see that the entire DNA molecule will have a negative charge due to its backbone being negatively charged. A small warning to those with trypophobia: you might find the following image unsettling, but here we have a gel called agarose gel. This gel is sort of like a jelly, except it's not as fluid, and it's basically made out of agarose, which is a polysaccharide, a sugar made of multiple monomers or monosaccharides. These different monomers join together to form a linear chain, and so we call agarose a linear polysaccharide. When a lot of these different chains overlap, then you get a sort of mesh-like structure, and that's what's shown over here in this scanning electron micrograph, which is a very, very zoomed in version of this gel on the right. You can see that these pores inside the gel, they're approximately 100 nanometers in diameter. So very, very small. Only like small molecules can pass through such widths. In fact, you might observe a similar structure in the red seaweed we saw at the start, because this agarose sugar is actually extracted from the cell walls of this seaweed. And that's why it's so important to the field of biotechnology, because it helps make this agarose gel. This is the gel that we use in gel electrophoresis, and we use it in a setup like this. So the blue part is the gel seen previously, and surrounding it is this tank called the electrophoresis tank. Now we make these small grooves inside this gel by basically cutting out a sort of cuboid from it, and inside these wells we fill in the molecule that we want to perform gel electrophoresis on. So in this case, DNA will be filled in these small wells along with a buffer solution. The thing about DNA though is, especially in eukaryotes, our DNA is very long. It's a huge molecule, millions of bases. It contains millions of bases. And so it wouldn't really fit inside these 100 nanometer pores shown above. So we use something called restriction enzymes, specifically endonucleases. Say you have a DNA strand like this, and it's continuing on both sides. Now, the endonucleases, endo basically means within, and nucleases are enzymes that are responsible to cut DNA. And so endonucleases are going to cut DNA within. So what they do is they travel along the DNA sequence and they look for specific sequences of bases. And let's say this enzyme is specific to this sequence. So once it finds it, it's going to splice the DNA over there. And in some cases, it will produce things called sticky ends, some cases blunt ends, but that's not very relevant here. So all you need to know is that these restriction enzymes are specific to certain bases, and once they find them, they will splice the DNA over there. So we can use them to convert our really long DNA into DNA fragments, small pieces of DNA, which can then be put inside the wells for gel electrophoresis.